So my name is Tjelvar, and I work at the John Innes Center. And how much lag is there? Well, there we go. So, okay, continue. 1 more minute, arranging my screen. There we go. So I started off my career studying biochemistry. So I did some science at one point, and that was at UCL. So I did an undergraduate and a PhD there, and that was very much on kind of structural biology, biophysics, looking at protein-ligand interactions, and the thermodynamics of that. It was really fun. Afterwards, I moved on and I moved to Cambridge, where I started working at the Cambridge Crystallographic Data Center. So that's I guess, the world's repository of organic and metal organic small molecule crystal structures. So if anyone's worked with the PDB, for example, so the protein data bank, it's very similar, but here we're talking about small molecules. So they have a very long history in data management. They started in 1965 with a woman called Olga Kennard, who had this revolutionary idea that if you collect all the data and make it available, then I guess like the sum of all the parts is going to be greater than all the little bits. So I think it's one of the first scientific databases that was ever created, and that's been going ever since. And I guess by the time that I was working there, we were making software and databases where we made use of that small molecule crystallographic data to make software for helping pharmaceutical companies, do molecular design or drug discovery, as well as distributing all that data to the whole of the academic community. Then in 2014, I moved to the John Innes Center. And when I started there, I was working a lot on image analysis. So a complete shift for me, really. I never really done anything like that before. And that was fun. I'm doing lots of project support, really good stuff. But around that time, I guess, next generation sequencing started taking off, and the John Innes Center was starting to accumulate more and more, both raw data as well as, I guess, derived data that came from the analysis of all that raw data. And we were running out of storage. And I guess at that point, my role changed into infrastructure and data management. And I was involved in developing lots of, I guess, protocols, as well as tools for managing all that data, and helping in terms of training people to make better use of the storage that we had available, make more efficient usage of it. And that training, I guess, was both like on an individual level as well as on a group level, and sometimes on project levels similar to this, maybe a bit smaller. And I guess it's in that capacity that I've kind of been thinking about data management, and I guess that's what I'm going to be talking about today. So I've given some disclaimers already about not being all-knowing. I'm not all-knowing. I don't know everything. I don't have all the answers. I think importantly, one of the things which I've learned from doing this is that every research group might not be completely unique, but they have different data that they're working with. They have different challenges. Different projects have different challenges. So it will kind of be up to you to have to figure out how you want to manage your data. And I guess what my role tends to be is help people think about the data and think about the challenges that lie ahead, and maybe give some suggestions on how they can tackle that. But I'm not here to kind of prescribe you anything or come with some kind of solution that just solves all the problems. Data management is quite hard. So in terms of, I guess, just getting you to start thinking about this, I think posing questions is a good way. So I guess one question is, how would data loss affect you? Or have you ever experienced data loss? What was that like? What would it be like? You wouldn't care, or would it be devastating? Would ruin your career, lose your job? I don't know. To what degree would it kind of upset you? So I guess that's one way of kind of getting some motivation for people to care about data management. I'm guessing you're all here, so you care about it a bit. at least.
Tjelvar, if you want, we can run a poll to see where
Yeah,
people are.
please. That would be great. Yeah, thank you.
Let me have a look here. So, oh, hang on a minute. I guess we've got this just simple one really, just to get a sense for people. So
yeah, great.
I think if people can fill that out, just a simple idea. Have you ever suffered data loss? I do have other ones here, but I'm not sure where they are. Somewhere. There we go. Can you see the percentages coming up there on the screen?
I can't. Do I need to stop sharing or?
Yes, it's just me. Hang on, I'll do it. So there we go, 30 seconds. So pretty even, actually. Let me.
Oh, wow.
There we go. So about half and half, I guess. So there we go.
Cool. So I'm guessing that the people who have suffered data loss, they tend to be the people that really start caring about data management. That's what I found in the past. So again, later on in the discussion, I'd love to hear some of those examples if you're willing to share them. I think it's useful as a kind of learning exercise for everyone. But again, if it's too personal or too sore, don't feel obliged. I guess a slightly more positive spin on this thing is how could data management make your science more effective? So I guess it's like the steak, and here I guess it's the carrot. Can you think of examples of how it might make your science more speedy, more efficient? I guess one example that we have at the John Innes Center is that people run out of storage quota when they're doing all the NGS analysis. And at that point, they get stuck. And it might take them a very, very long time to free up some space or to work out what they can and can't delete to move on with their analysis. So that's something where I guess if you manage data, it can be simpler, and then you don't have to have this kind of massive delays because you run out of storage quota. This example here, I guess, is more really an example of how data visualization can be effective, but I think it's also a case of data management. I'll take that. So this is John Snow, who traced the outbreak of cholera, and I guess he managed that data, and then he mapped it onto a map to trace the pump where that cholera outbreak came from, and started this field of epidemiology. So yeah, data management is a magic. It makes you do better science. Yeah. I guess the other question to consider is what types of data will you be generating? And I think as you guys were describing what you're going to be doing, I think it's a little bit of everything. I heard a lot of next generation sequencing, so that's a picture there on the right. I heard some imaging as well, in terms of, was it chloroplasts or something like that? Different challenges with the different types of experiments and the amounts of data that they generate. But also I had people doing field trials and doing phenotypic collection, or collecting phenotypes and stuff like that. And I guess depending on what kind of phenotypes you're measuring, you might be out with a ruler, or you might be out collecting something else, and then I guess recording that in your notebook, and then I guess at some point transcribing that, or maybe you're writing it straight into your iPad. But these have different challenges. And I think maybe I heard some of you also talking about metadata, and it was really nice to hear. So what metadata will you need to make sense of your data? So I guess metadata is data about data. So say, for example, that you've done microscopy and you've got an image of some cell. In order to make sense of that, you need to know maybe, what's the species, what's the tissue, what's the stain, is it a control, is it a mutant, all this kind of stuff that isn't actually recorded in a microscopy file. That's stuff that you need to know, and that's metadata that you kind of need to keep track of and associate with that file. All that stuff that you record in your lab notebook is potentially metadata. How are you going to associate it with the files that you're generating? Yeah. So that's something to ponder, and I'll come back to this later on. So as I mentioned, I was talking to lots of groups at the John Innes Center about the data management to try and help them work out how they can do that. And I guess there were some principles that kind of emerged from that. And the first principle, make it clear who's responsible for what. Yeah. So like I said, this is a very high level view of data management. And I guess is to illustrate what I mean by that, who's responsible. We'll make a fictitious example here. So we have ambitious Anna, who's a group leader, and fastidious Fatima, who's an experimental biology postdoc, and binary Beatrice, who's a group bioinformatician. So I guess in this example, Anna and Fatima, they're talking about a particular mutant, and they want to analyze it. It's got some phenotype, and want to try and see if they can work out the molecular basis of that. So they decide that Fatima should do some next generation sequencing to see if she can discern what the difference are in the expression levels. So Fatima, she goes away, she grows the plants, she takes the samples, she extracts the RNA, she sends that off to some sequencing company. And 2 months later, she gets the results back. But she doesn't actually get the results. She gets the link to a download, and it's got nasty instructions, and you need to download specific software to work with that download. So she doesn't really want to struggle with all that stuff. But she knows that Beatrice has done this before for her friends and colleagues in the same lab. So she goes and asks Beatrice for help. Beatrice says, oh, gosh, not again, you know, what am I? Like, you know, the person who downloads data. But she says, yeah, sure, I can download it. So she downloads it. And then later on, she has another chat with Fatima and realizes, you know, actually, you know, Beatrice should do the analysis as well. So Beatrice does that. And I guess in this scenario, then, who is responsible for what, or who's responsible for that raw data? So let's not involve the derived data at this point. Let's just talk about the raw data. So Anna, she has never seen the experimental samples. She's never seen the files. She thinks that the people in her group are responsible for managing that data. Fatima, even though she managed the samples, she never downloaded any data. So she thinks, you know, Beatrice downloaded that, so she's responsible. Beatrice supports, I don't know, 6 other postdocs in the group. So she, and as well, she's going to need to find another job in a month's time, or in 6 months' time when her contract runs out. So she doesn't think that she's responsible for the data. She thinks, you know, this is all Anna's data, so Anna is responsible for the data. And I guess in this kind of tenuous example, no one actually takes or thinks that they are the person who's responsible for the data. So I guess the point that I'm trying to make here is that you need to have a conversation within your project or within your group to try and work out who is responsible for what. And I guess what I've written here is something that came out when I helped another project working on this. This was also kind of cross-group project, where they came to the conclusion that ultimately the data management is the responsibility of the group leader, but the group leader is unlikely to actually work with the data. So they're going to delegate this responsibility to a data champion. And then the data champion is going to be responsible for ensuring that existing new members of the group are aware of the group's data management processes. Yeah. So like I said earlier, I'm not trying to prescribe this to you. This is an example of how one project decided on a process for managing the responsibility. Yeah. If you decide to have a data champion, you can have a challenge of finding someone to volunteer for that.
This time, should we ask to see people's status with regard data champions in their
That
groups?
would be fantastic. Thank you.
There we go. I realize, of course, a lot of these projects are just beginning, but do you know who it is who would be in charge of the data that you generate? Almost seems like we prepared this beforehand. Okay, almost there. Just to give it a couple more seconds for people to vote. Okay, so we've got most people have shared. So let me share the results there. So I think that says a lot. I mean,
Good.
this could reflect the status of many of these projects, of course, but
No, I think it's. I think that's encouraging. That 30% has it is good. Cool. Great. So at that point, the second principle. So this is, I guess, where this talk takes a slightly kind of bioinformatics-y turn. But it doesn't need to be exclusive to bioinformatics. I'll try and show how it can be relevant to other stuff as well. But there's this concept of keeping raw data safe. So that's relatively, I guess, common knowledge. I'm hoping that you want to make a backup of your data. I'm sure most of you do that already, or hopefully. In terms of keeping it safe, I guess something else that you can do is you can change the permissions on those files to make sure that they're read-only, so that no one can kind of accidentally delete them or overwrite them. I think that's probably less common, but it's something that you can do. But then there's this concept of keeping it separate from derived data. So what do I mean by derived data? I mean data that, I guess it's not the actual raw files. It's, I don't know, if you've run a bioinformatics script and maybe you've generated a BAM file, or maybe you're working with a microscopy experiment, and in that microscopy file, you have actually 5 samples that you've imaged. And for a presentation, you start, I guess, or whatever, for some analysis, you start extracting bits of those files and saving them as, I don't know, PNGs or TIFF files or something like that, because you think that these are kind of good examples of what you want to illustrate. I guess that derived from that raw LIF file or CIF file. So that's what I mean by derived. You've done something to get it, and you could potentially get it back if you went through and did the same steps again. Okay, so as long as you have the raw data, you could get the raw data back if you have the right instructions to do so. So to show why I think this is important, or why it can be difficult. So here's a fictional example as well. Again, you can see that this is a slight bioinformatics slant here. But we've got lots of, again, .py files, so the Python, the script files, essentially. You got a bunch of CSV files, which is, I guess, like spreadsheets. And then that's it, okay. But we don't really know much more about this. And I guess at the John Innes Center, we ran into this problem that actually we didn't have enough storage. So people had to be able to clean up, the derived data, the data that maybe the data that wasn't the analysis that hadn't been meaningful, or stuff that they could regenerate, just to free up space so that they can do more analysis. The situation is better now. We've kind of resolved it. We've got more storage, but there was a period of at least 2 years when I kind of came in to start helping out, where we didn't have enough space and people had to really manage what they were doing. So I often came to this type of scenario where I've looked at people's directories and said, like, oh my gosh. And it was a lot worse than this. Yeah. It's just a big slew of files and you don't know what's what. So why is that? Why do you get to that situation? So it's quite easy to get to that situation, I think. So here, hopefully you can see my cursor, but I'm starting up in the top left corner, and you've gotten some data. And I guess the natural instinct is to be curious. So you want to see, what does that data mean? So you start delving into it, and maybe you write a script to analyze that data, to make a plot or whatever. And then this is, I guess, where it's, especially when you're new and you're doing informatics, you're doing bioinformatics, dealing with paths and saying, you know, how do I point my analysis script at the raw data? And the absolutely easiest thing to do is to stick the analysis script next to the raw data, because then you don't have to worry about relative paths or where on the computer this file is. You just know it's right next to it. I just need to provide the file name. And then you run that script, and that probably produces some output. So that's down out and here in the second little box, if you generate some output. And still, it's relatively simple. At this point, you're not worried. It's kind of this frog that's being boiled. You know, you don't really sense that the temperature is increasing, so you don't jump out. You just continue. And at that point, some more, no, actually, you realize, oh, there's a problem with some of the rows in this file. I need to do some pre-processing to, I don't know, maybe strip them out. So you're good about it. You write a script to do it so that you can reproduce it. So now you have a script that does pre-processing, that creates a clean data file. And then you run your analysis again, and that produces some output again. So this has been regenerated now. But you see, it getting a bit more busy. And then at that point, you get more data, yeah? And you realize, oh, actually, I need a script to analyze things from more than one input source. And then you rerun it and you regenerate the clean data and the output CSV. And all of a sudden you're in this situation where I showed you in the beginning, where things are a bit messy. And if you're at that point you need to clean up, it's very difficult to kind of work out what's what. So imagine a different scenario where you upfront think a bit more about the organization of your files. So you start off with creating a directory for raw data, because you know that's raw. I want it to be pristine. I don't want to touch anything in there ever once it's there. And then maybe you create a separate directory or folder for scripts. So you know these are the stuff that I'm going to use to analyze things. And then what I would suggest is that you create another directory for intermediate data. So here, this clean data, when we run the pre-processing, that's intermediate data. Yeah, so we can regenerate that, or maybe we will need to regenerate it often because we get more and more raw data. And actually we need to regenerate that frequently. And then possibly you want to separate out final results. So maybe it is the case that, you know, you're going to publish something and you want to make sure that the output.csv file, the file that you had to make your figures and stuff for the paper, that's something that's separate. That's something that maybe you don't want to delete that. Maybe you want to keep that. But the intermediate data is kind of, it's still in this kind of transitory state, but maybe something that you want to keep for a longer time, but it's not really raw. So again, I'm not here to prescribe what you do. I'm just saying, like, I think it's a good idea to kind of get a bit more granularity and separate out raw data and scripts, and then something else where for the output of your scripts. So these scripts are going to point at the data stored somewhere else, and it's going to write to a direction somewhere else. So it's not all happening in the same place. Okay, brief. Principle 3.
Well, should we ask about raw and derived data at this stage? So
yeah, please.
There we go. If there's to launch the poll about whether you've got an awareness of keeping that raw data from the derived data, or whether that's something everyone's kept kind of all together. Owen? Okay, so most people have answered, and I think this will make everyone happy.
Cool, fantastic. That's great.
Good sign.
Good. So then this third principle is about, I guess, a slightly bigger picture than the nitty-gritty of individual files. It's about standardizing the location, but also the structure of data. So why do I talk about this? So I think, depending on how your group works, but say that you're a PhD student or you're a postdoc and you've worked really hard to grow some plants, extract RNA, or you've taken microscopy images, or you've been out measuring phenotypes and recording them into some spreadsheet. You've put in a lot of effort, and I think to you, you feel this is my data. I worked really, really hard for this. You know, this is 9 months, 2 years worth of work. It's mine. It's my blood and sweat and tears. Yeah. But is it really yours, or is it the group's? Yeah. Is it the group leader that actually owns that data because they paid for you to do that work? Or is it actually the funding body's data because they sponsored the group leader to ask you to do that work? Yeah. Maybe that's a slightly separate discussion, but I think if you don't have a kind of standardized way for talking about how you're going to structure that data, then I'm going to go away and do what I think is best. Yeah. And maybe I think it's best to do things in a certain way. And then the other PhD students and the other postdocs, they're going to go away and they're going to do what they think is best. And all of a sudden you've got, I guess, lots of people doing different things. So maybe Anna is organizing her data by grant codes. And Fatima, she's organizing her data by the date that it was collected or generated. And Beatrice, she organizes her data by species. It's very easy for every member of a group to kind of start inventing their own way of structuring the data. And it makes perfect sense to them, but that doesn't necessarily mean that it makes sense to anyone else. And then when someone leaves the group, it's very easy for that data to become incomprehensible to anyone else, or that it takes a lot of effort for someone else to understand it and organize it, make sense of it. So I think the point there is to have a discussion. And I guess this also then comes back to this idea of, well, maybe you do want to have a data champion, maybe you don't, but at some point you need to have a means of communicating what that standard is. So here's an example. And in this example, you're going to have to just imagine that in this project, in this readme.txt file, are all the instruction that details how data should be managed. So when someone comes to this project, the first thing they see is this readme file and they read it, hopefully, or you can tell them, go read this file. It will tell you everything that there is to know about the data management. And that file says raw data should be stored in the raw data folder, okay? Everything goes in there. And then maybe you have work packages, maybe you have projects, maybe you have people, maybe you have analyses that you think are interesting, but you store individual bits separate. So this comes back to this earlier. You have scripts, you have intermediate, you have final results. And these scripts, the first thing that they do is either like they point at the raw data, or maybe if the files are small, it actually copies the raw data into the intermediate data here. And then it continues to work on them and generate some final results maybe. Yeah. So the level that you use here, it's up to you. You decide what you want. I mean, here are work packages, but that could be projects or it could be individuals. But I think the value of this is that if you're the person that generates a piece of data, you've done that for a purpose. You wanted to do some analysis. You wanted to answer a question, yeah? And that's fine. So maybe you think that that data should live in your project folder under, you know, this question that I wanted to answer. But when another PhD student comes along after you leave, maybe they want to use that data for another purpose. And then what do they do? Should they take a copy of it, or do they continue to work in your folder and go on from there? So I think that's why it's useful to kind of keep the raw data separate from the individual projects or people who generated them, just because that data could be useful for other people. And if the people that need it reference that raw data in a unique location where everything is stored, I guess it's just, to me, it's much easier to kind of keep track of what's going on. But again, I'm not here to prescribe, but I guess the problem is if everyone is in charge of their own pieces of data, then when you get someone who comes along and wants to make use of lots of different pieces of data from many people, it's more difficult. That's that for principle 3. Principle 4, provide metadata. Okay, so up until now, principle 1, 2, 3, I think that they're relatively easy to grasp and they're relatively easy to implement. Principle 4, when it comes to providing metadata, I think this is when it gets more complicated. Yeah. And there you kind of, it's difficult because I don't think that there necessarily are great tools to do this. Yeah. So one solution that many people use is to store the metadata in file names and directories. So here's an example. And okay, so here I'm using a Linux kind of Mac convention of directories where the forward slashes are denote directories. If you're on a Windows, imagine that those forward slashes are backward slashes. Yeah. But the first directory level says that this is the date that I did this on. The second directory level describes the accession. So I guess calls here a wild type of Arabidopsis. And the third nesting of the folders or directories is the replicate. And then you've got the file name, which describes the treatment. You know, it was a control in this case. And then actually the file extension means something too. It means that actually this is CSI file, so it's size microscopy image. That's good in that's easy to use. But there are some disadvantages in terms of this being a bit fragile. So there's a risk of losing metadata if you're renaming a file or if you're reorganizing the directory structure. You do lose metadata if you just copy the file alone to a different location or you send it to a collaborator, because that file doesn't really make any sense now without a directory structure. And another thing, it's quite difficult to get an overview of the data because you have so many levels of nesting that it's difficult to get a kind of eagle-eye view of what you have. So an alternative approach is to store metadata in Excel files. So here, I guess you have a column for every piece of metadata that you want. So you have, in this case, you have a date, an accession, a replicate, a treatment, an experiment type. It's good, it's a bit more explicit. It says what each column is meant to be. And then at some point you have also then a column for your file path, which says, where is the file that's associated with this metadata? And again, it's relatively easy to use. And here now it's easier to get an overview of the data that you have because you can look at that spreadsheet and I guess you can filter it or whatever you want to do with it. So you can get an overview. But again, it's got some disadvantages in that it's very difficult to move files or reorganize the directory structure where you have the raw data, where you have the data files without breaking the links in the Excel files. Yeah. So you get scared, it gets difficult, or maybe you do it and maybe you do lose the link. So maybe it is that you don't want to do it, but you accidentally do it, or maybe it is that you do want to do it, but you feel like you can't because you're scared because you're going to break things. Again, there's a loss of metadata if the file alone is copied to a different location or if it's sent to a collaborator. And I guess with Excel files, it can be difficult to work with from command line. So again, maybe you're not a command line person, but maybe you are. Maybe it's something that you want to do. You want to be able to access metadata from the data. So I guess this was something that we were struggling a bit with, and this is where we implemented some tooling to help us manage data. And I'm not going to go into deep details of what we did. If you're interested in this stuff or you think it sounds useful, I'm quite happy to talk to you separate from this meeting and help you get started with this thing that we created. We call it D-Tool. It's meant to be very lightweight, so it's not meant to be prescriptive. Essentially what you do is you create a box and then you stick your data in the box, you close the box, and then you add metadata as a kind of label or sticker on that box. And then when you're working with that, you're not working with the data anymore, you're working with the data set, that's the box, and then you have access to the metadata as well as the metadata as one thing, you know. And I guess initially it had a couple of advantages for us because it meant that it was easy to move data sets around without losing metadata. So we had an issue that we were running out of, I guess, our high performance storage where people were doing their analysis on the cluster. But then we asked people to package the data up as data sets, and that meant that they could move them because we had some other storage. But it was cheaper storage, it was object storage. And this D-Tool that we wrote abstracted away the underlying storage system and meant that we could just copy the data into this cheaper storage. And again, what you get inside of that cheaper object storage is both the data and the metadata as a unified whole. So we didn't have to worry about this kind of idea of metadata loss. The other thing, which I haven't really touched on yet, is that there's different types of metadata. You can also have what's called structural metadata, which I guess it can, so an example would be like the size of a file, yeah? Or another thing is the checksum of a file. So you might see this if you've downloaded sequencing data from a sequencing provider, they provide these MD5 sums. So that's basically a short piece of text that you get when you run this MD5 sum check on a file. So it's meant to be unique for every file depending on its content. So you can then run that on the file that you've downloaded and you can check that checksum that you get against the one that you were expecting. You can see that the file is actually got the content that you expected it to have. So we built that into the tooling that we have. So it's very easy to check the integrity of the data. Again, it was kind of associated with this idea of copying data to somewhere else, like this object storage. Did it actually work? You know, there was this kind of hesitation because these were new technologies that we were working with. We wanted to make sure that everything was sound, that there hadn't been any kind of corruption. So I guess in this term, D-Tool, what we wrote, it's got some advantages. I think it's easy to move data sets around without losing metadata. It's easy to get an overview of the data. It's easy to verify the integrity of the data, and it's easy to use from the command line. But I guess the disadvantages are that it requires you to learn how to use D-Tool. Yeah. So I think people know how to work with Excel, people know how to work with directory structures and file names, but using a separate tool can be a bit of a, it's not something that you know already. And I guess initially it only had a command line interface. Now I'm working on making a GUI for it, but it's still being developed. I mean, it's kind of beta stage, but I wouldn't say that anyone should go and use it yet. Anyway, I'm not going to go into details here too much. If people are interested, I can talk about it separately. I'm not going to try and force that on people. So in summary, I guess make it clear who's responsible for what. Keep raw data safe and separate from derived data. Standardize their location and structure of your data and provide metadata. So here are some links to further reading. So I think you can have all these slides afterwards. So this talk was mainly based on this blog post I wrote a while ago, Data Management for Biologists. Another good resource for describing how to work with, I guess, digital files is this 10 Simple Rules for Digital Data Storage by Hartdell. Leek has written a really nice blog post about how to share data with a statistician. He keeps banging on about how to keep the raw data raw and also provide metadata. And then I guess this last one, I guess you don't really need right now because you already got your funding, but I guess it's about how to write a good data management plan. Might be useful for you if you're coming up to new projects that you want to get money for in the future. So with that, I'm gonna finish this talk. That's my email at the bottom. If you wanna get back to me, if you have any queries or whatever, I would love to get feedback. Was this useful? Or what was useful? And then I guess, what do you wish I would have done differently? So in case I do this again to some other project group or group of people, it'd be useful for me to get feedback on what worked and what could be improved. So thank you very much for listening.